I'm not a synthetic biologist, although I find the topic extremely interesting, and I do believe that there are important applications of the field to cancer research. And we do a lot of genetic engineering technology in my lab, and actually my purpose today is to expose you to some of those technologies that we use routinely, and I think they will be of value to the synthetic biology community, and I can imagine lots of collaborative opportunities, not just with my own group, but with others that do the kind of work that we do. So, as all of you know, cancer is a genetic disease. Normal cells acquire mutations in cellular genes, oncogenes and tumor suppressor genes and other genes, and as such become converted into cancer cells. And there's a great interest nowadays in understanding the full complexity of the genes that are altered in cancer. And as you've been reading about, there are many, many genes that are altered in at least human cancers. Tens or hundreds of genes get mutated over the course of years and decades in the development of a cancer. And if that weren't complicated enough, cancer cells don't sit alone. They sit in the body and interact in important ways with other cells, including cells that try to inhibit their growth, like cells of the immune system, and other cells that promote their growth, like cells of the vasculature. And so there are very significant cell-cell interactions that take place during tumor development. And a fine example of that is the movement of primary cells in the process of metastasis to distant sites in the body, where the cells move through the lymphatics or the blood system and take up residence in some other tissue. This, of course, is the lethal phase of cancer. 90% or so of cancer deaths are attributed to this phase of the disease, and it's actually one that we don't understand very well. So there's a need to understand the genetic basis of this quite well. But it's for the reasons that I just reviewed, the fact that cancer cells arise in context, that for some years we've been studying tumorigenesis not in cells in a dish, but in whole animals. And we're not alone. There are many such investigations going on. In fact, there are many model systems that one can use to study tumorigenesis. We focus on the mouse, but one can also do these kinds of studies in zebrafish or in rats or in invertebrate systems like C. elegans and Drosophila. We focused on the mouse partly because of the long-standing history that exists in genetic engineering in the mouse, which dates back more than 30 years. We can control the genetic background of the animal, so we don't have to worry about confounding variability between the ground state. In addition, we can grow these animals up to large numbers in the laboratory and monitor them very closely. So that's the reasons that we've focused on the mouse over the last now 20+ years. Most of you are familiar with the kinds of methods that we use to modify the genomes of mice. So I'm not going to belabor this point, but for those of you who aren't, I thought I would at least review it quickly. One can, of course, construct standard transgenic strains using, for example, a tissue-specific promoter driving a particular oncogene to express that oncogene in a cell and hope that it becomes transformed. That was the old-style method of making mouse models. More recently, one can knock in mutations of interest into the cells, into the cellular copy of that gene, and thereby model more closely what happens in real human cancer. And you can do that in a constitutive fashion where the gene is just altered by, for example, a point mutation, or you can go a step or two further and regulate the expression of that mutant copy of the gene. For example, using a conditional strategy, and I'll show you one that we use in my lab where we introduce a stop element, a transcriptional stop element, to silence the gene until we want it to turn on, and then we can activate the gene at a time or place of interest. And you can also do that in a regulatable fashion using, for example, doxycycline systems, using TET repressors that have been modified to be activated by binding to doxycycline. You could introduce these elements into cells and then using DOX addition or DOX withdrawal, turn the gene on, allow a tumor to form, turn the gene off, and ask what happens when that signal is removed. So these are the kinds of gain-of-function mutations and tricks that we use. There are also loss-of-function mutations and tricks we can use, including standard gene targeting technology where you inactivate the gene in the animal and establish the consequences of that mutation. And then second and third generation versions of that where you use, again, conditional strategies like Cre-lock strategies where you can delete the gene using Cre expression or some other recombinase, again, at times or places of interest, using either tissue-specific expression of Cre or introduction of Cre or other recombinases on a virus, and I'll show you examples of that as well. So we and others have built lots of tools like this. Any gene you're interested in cancer has been manipulated in the ways that I just described, and you can look at those in isolation or you can combine them together and look at them in combination. And as such, one can construct genetically engineered models and initiate the tumorigenic process in one way or another and then watch it unfold in ways that you can't do in people. In a person, you, of course, are presented with the fully blown disease. All that stuff has already happened. Here, we can sort of light the match and watch it burn and then try to study the process as it goes forward. And that's the main focus of my lab, actually, is to understand the detailed steps of tumorigenesis. But once you have these models, you can also use them for a variety of practical purposes, like how do you treat the disease better with chemotherapy or new anti-cancer agents? How do you prevent the disease from happening in the first place? That's a synthetic biology opportunity right there. And how do you detect it earlier? Obviously, if we can find cancers at earlier stages, we can radically improve the treatment outcomes for those patients. And so we hope that these systems will be valuable in that respect as well. And again, I think there's a synthetic biology opportunity there. So the disease that I'm going to focus on primarily is lung cancer. It's the one that we study most extensively and have done for about a dozen years. Lung cancer, as you know, is the leading cause of cancer deaths in the US and around the world. The survival rates are not good, and it's partly due to the fact that the disease tends to be diagnosed at late stage once it's already moved outside of the lung in metastasis, so the patients don't do very well. And that raises two important points. One is better therapies, of course, and also to understand this progression process so that we might be able to interfere with it. And also relevant to this talk, we've known actually for quite some time that there are two very major players in so-called non-small cell lung cancer, especially the adenocarcinoma type of non-small cell lung cancer. That's the most common type. That's the one that we study. And it involves mutations in KRAS, a very important human oncogene, and P53, a very important human tumor suppressor gene. And so based on that, we built our model to activate KRAS and inactivate P53 in hopes that that would lead to advanced non-small cell lung cancer in the animal. And so we constructed the so-called lock-stop-LOX allele of KRAS. This allele has an oncogenic point mutation in codon 12, like you would see in human cancers, knocked into the endogenous gene. But the gene is silenced by virtue of a transcriptional stop element, and the stop element is surrounded by LOXP recombination sites, such that in the presence of Cre, the stop is removed and the gene turns on. We also got from Anton Burns's lab a floxed P53 allele, so this gene is wild type in the germline configuration, but it can be inactivated using Cre recombinase. And in this case, we use viruses that carry Cre, adenoviruses or lentiviruses. The animals inhale the virus then infects cells throughout the lung. When they infect the proper cell, RAS gets activated, P53 gets inactivated, and that initiates tumor formation. Animals will develop primary tumors within about 4 or 5 months in this context, and a few months later they will develop distant metastases. So indeed, we can develop models of this advanced form of human lung cancer and then study it in a variety of ways. The lentiviruses are quite useful, not only to introduce Cre, but you can also bring in other genes. And here you should be thinking about what you would do. You can bring in other genes or other modules along with the virus. For example, you can bring in shRNAs or microRNAs and then deliver them directly to the cells that are going to become the tumor. And that all of the descendant cells will inherit that virus, so you'll see that the properties of that expression in all the tumor cells. You can also bring in genes via cDNAs into those cells, therapeutic genes, imaging genes, and so forth. For example, you can put GFP or RFP in here so you can light up that tumor as it's developing. And that's illustrated here in a very simple example, a GFP-labeled tumor cell that allows you to visualize the tumor very easily, follow it very easily, see it go into the blood, for example, and so forth. So we can manipulate these cells very precisely, and the modules that you guys are thinking of building could be easily modified to be placed in this context and studied in an evolving tumor. The tumors do evolve from early stages, from single cells through hyperplasia to adenoma formation and advanced adenoma formation. And then eventually, in the context of a p53 mutation, will advance to adenocarcinomas, and some of these will progress, as I told you already, to metastasis. Now, the mutations occur at the same time at the beginning, activation of RAS and inactivation of p53. We actually believe that the p53 mutation is irrelevant up until this point here. Plus or minus p53, you mostly make the same type of tumor. It's relevant here in the transition from an adenoma, a benign tumor, to an adenocarcinoma, a malignant tumor. So we actually believe that p53 is regulating this transition. And that's based on a lot of work that we've done over the years, including recent work from David Feltzer's lab, from David Feltzer in the lab. He now has his own lab at the University of Pennsylvania, using a method which I'll now introduce you to, where we can turn p53 back on and ask what happens. And it's based on those observations that allowed us to place the p53 step right there. Now, some of you work on sensors, and I would argue that the coolest sensor I know is p53. p53 is a master regulator of cellular stress or response to cellular stress, and there are many inputs into p53 and many outputs from p53. On the upstream side, the activators of p53 are, for example, DNA damage, induces phosphorylation of p53, which leads to its stabilization. Oncogene activation, likewise, through a different mechanism, can activate p53. But other things, too, like hypoxia and general genomic instability can do this as well. So there are various inputs into p53 that lead to increased levels of the protein. The protein is a transcription factor, goes about turning on target genes, and then instructs the cell to either arrest its growth, either transiently or permanently, perhaps to fix the damage that has occurred, or undergo apoptosis, a more significant cellular response. And there are other things that p53 will coordinate as well, like effects on angiogenesis or damage repair. So p53 is an important gene. p53 is inactivated in 50% or more of human cancers. And so we wondered what would happen if one could restore p53 function in a tumor. And we had the tools to do that, so we asked that question. Here's the pathway in a more simplified form. As I've said, p53 is inactivated in the model, of course, but also in many human cancers. And upon inactivation of p53, one eliminates the cell cycle arrest and senescence responses. So the question is, what if you put p53 back in the game? Would you restore cell cycle arrest and apoptosis responses, which would be a good thing? Or is it too little too late? Perhaps it doesn't matter to a fully established tumor cell that you restore p53 function. Maybe all the relevant stuff has already happened, and the cell couldn't care less if you put p53 back on. But that's the question we wanted to ask, and here I'll introduce you to another genetic tool, and that is a restorable allele of a gene of interest, in this case p53. So we use the same lock-stop-lock strategy, but not to control the expression of a gain-of-function mutation. This by itself is a loss-of-function mutation. It inactivates the gene. So in the germline configuration, this looks like just like a p53 null allele. It's non-functional. You don't make any p53 protein. Again, the stop element is surrounded by LOXP sites, so that in the presence of Cre, the stop is removed, the gene turns back on, and it's just the old wild-type gene. So we can control the activity from off to on. We can flip the switch in that direction and ask what happens. We need Cre for that, and we want it to be activated in all of the cancer cells and actually other cells too. And so we built a Cre allele that's fused with the estrogen receptor and is therefore tamoxifen sensitive and is expressed from the ubiquitous ROSA26 promoter. So all the cells roughly in the animal express ROSA26 Cre-ERT2. You add tamoxifen, Cre gets activated, you remove the stop, and p53 comes back on. Now, animals that are homozygous for this mutation and carry this allele are cancer prone. They will spontaneously develop cancers, lymphomas, and sarcomas. And so Andrea Ventura in my lab first looked at the response of those tumor types to p53 restoration. And here you can see a lymphoma, an abdominal lymphoma in one of these animals by MRI imaging. And here's a large sarcoma in a different one of these mice. Again, these happen spontaneously because p53 is inactive. We can then add tamoxifen, wait about a week, p53 gets reactivated or restored, and then we ask what happens. And in these cases, it's quite dramatic. The lymphomas shrink away to almost nothingness. Apoptosis is activated, and the tumors die away. In the case of the sarcomas, it's a little less dramatic, but we still observe shrinkage of these tumors. It's not by apoptosis. These cells induce senescence. And then some cell of the innate immune system, we think, will clear those senescent cells away. So once again, we see an anti-tumor response. So indeed, restoration of p53 function has the desired effect. The cancer cells do care, and they are eliminated. This is true also in the lung model that I told you about. David Felser showed that indeed, if you restore p53 in the KRAS p53-driven model, you go from having these large tumors, visualized here by micro-CT imaging, to much smaller tumors. Before p53 restoration, the tumors would look like this. After p53 restoration, they look like that, sort of a Swiss cheese appearance, and that's because cancer cells are eliminated. Interestingly, it's not all the cancer cells. It's only the advanced cancer cells. It's only the carcinomas. The adenoma-like cells, the earlier stage cells, are spared from death, or elimination in this case, because they don't actually activate p53. There's something about the carcinoma state that turns that circuit on, activates p53, and eliminates those cells. And we think we know what it is. We think it's oncogene activation. KRAS, which is driving tumor formation here, gets amplified right at that point. And that probably stimulates the p53 pathway, specifically in the carcinoma cells, but keeps the adenoma cells untouched. So that's an interesting observation. It's an important piece of biology, and it's relevant to therapy, but it's not going to be a therapeutic. Maybe we can get gene therapy to work well enough to introduce the p53 gene, which has been tried, and actually it's FDA, the equivalent of the FDA in China. It's approved in China. It's been tried here, but without as much success. Maybe it will work one day, but we decided to take a different approach to using this same strategy of trying to restore the p53 pathway, but not with p53 itself. So this is the second vignette I will tell you, and there are a total of 3 such vignettes. And that is to use a downstream component of the p53 pathway to try to restore p53 function or components of p53 function in a tumor. I told you that p53 is a transcription factor. It regulates a number of protein coding genes that are relevant to cell cycle arrest and apoptosis and repair, but it also regulates other types of genes like microRNA genes. There are microRNAs that are downstream of p53. Actually, there are link RNAs that are downstream of p53 as well. But for this, it's important to know that microRNA 34 family, miR34 A, B, and C, are transcriptional targets of p53. And it's thought from work of others that this is an important part of the p53 response. miR34 inhibits genes that are important in cell cycle arrest, in cell cycle progression rather, important in apoptosis inhibition, so that if you inhibit those genes, then the cells will arrest or the cells might die. So we wondered whether miR34 could be used to replace p53 in a p53 deficient tumor. And this is the work of Wen Zhu in the lab. And he did this first genetically. And here's an example of being able to use these lentiviruses. Here the lentivirus carries Cre, but it also carries miR34 downstream of a reporter, actually, M. cherry. The cells also have a RTTA driven by a lung-specific promoter. So when you introduce this virus into the KRAS p53 model, RAS gets recombined, p53 gets recombined, but now you've got the miR34 plugged in, and it can be induced in a DOX-dependent fashion. And we can do that either, we can induce miR34 either at the beginning of the process, right at the time of tumor initiation, or we can wait, allow a tumor to form, and then turn miR34 on to ask what happens in an established tumor. So when Wen did that, first at the time of tumor initiation, he saw a very dramatic effect. This is the control situation. Absence of miR34 induction, you get nice tumors. If you induce miR34 at the time of tumor initiation, you don't get any. miR34 blocks tumor initiation, probably by inducing arrest or death of the cells that are trying to be initiated. That's interesting, but it's more relevant to ask what happens in an established tumor. And so Wen did that by waiting for tumors to form, imaging them by micro-CT, and then adding dots at that stage. And you can see that in that case too, in contrast to the control tumors that continue to grow, if you induce miR34, you find that the tumors stop growing. Stasis. And that's specific to the miR34 and isn't seen with a scrambled version of the miR34 microRNA. So that's all well and good, but it's still a genetic experiment, not likely to work in humans anytime soon. So can we convert this to a drug-like treatment? Can we introduce the microRNA therapeutically? And for this, we turned to our colleagues in the Koch Institute. We work with several of our engineering colleagues who are working on various delivery methods for small RNAs and other things, including Bob Langer, Dan Anderson, and Sangeeta Bhatia. Dan Anderson's lab has made a number of materials that have interesting properties for small RNA delivery. And we teamed up with James Dahlman in Dan's lab to test one such material. And I should say we've done other similar work with Sangeeta's lab. But this is a material they call 7C1 that they made using a combinatorial chemical engineering method. And they'd known that it had good delivery properties to endothelial cells in the lung. So we asked whether they might have good delivery properties to epithelial cells and cancer cells in the lung. And we did a series of experiments testing that question. First, control experiments using simply fluorescently labeled small RNAs. Could we deliver them to the lung and to lung tumors? And these data simply say yes. You can find fluorescently labeled small RNAs in lung tissue as well as, importantly, in lung tumors. We could also silence genes within those cells. Using the same KP model, we introduced two Cre-activatable reporters, luciferase and tomato, and asked whether we could silence the luciferase within those tumors. And the answer is yes. You can see silencing of luciferase following a single dose of luciferase siRNA. And that was specific. You could see inhibition of the luciferase signal, but not general inhibition of anything, because the RFP signal, which shouldn't be affected, isn't affected. So we can deliver small RNAs to these tumor cells. What about miR34? And indeed, we can deliver miR34 as well. Here, a single dose tail vein injection in this nanoparticle formulation. We can observe miR34 in the tumor. This is the one we delivered, and we don't see a control signal. And we can actually also see inhibition of miR34 targets within the tumor cells, even after a single injection. And the most important thing, is there a therapeutic response? And for this, we did multiple injections, tail vein, over the course of a few weeks. And indeed, we saw that the introduction of miR34 mimic by tail vein injection through this formulation caused, again, tumor stasis, a block in tumor progression, a therapeutic effect. And this therapeutic effect was due to induction of cell cycle arrest, presumably due to the inhibition of the targets that miR34 controls that are relevant to the cell cycle. So the point of this story is that, yes, you can do small RNA perturbations. You don't have to do genetic engineering. And I think that opens up all sorts of possibilities for analysis in vivo. OK, my last story, which I'll get through in a few minutes, has to do with this progression pathway, and specifically the last step, and that is the progression to metastasis. I don't have time to go through all the background here, but Monty Winslow in my lab looked by gene expression analysis at genes that are changed in their expression as tumors move from pre-malignant to metastatic. And he focused on one transcription factor called NKX2.1. It's a homeobox transcription factor, which is important in lung development and goes from being expressed in the tumors to being inactivated, inhibited in the tumors, expressed in early stage disease, not expressed in later stage disease, in the metastatic stages of the disease. These are cell lines, but you can also see this in tumor tissue. Early stage disease, well-differentiated cells express this transcription factor. Later stage, less well-differentiated cells lack the transcription factor. They do become less differentiated. It's not too surprising, but we wondered whether that gene, that transcription factor, was changing the biology in other interesting ways. This transcription factor is very important in early development in the formation of the lung. As Doug Melton told you about before, many of your internal organs come from the gut tube. He told you about PDX1 and the formation of the pancreas. NKX 2.1 is important in the formation of the lung at a few days later in development in the mouse. You need NKX1 to turn from gut to lung. And in our case, we're forming a lung tumor, which is itself expressing NKX 2.1. And the question we wanted to ask was, what happens if you took an end-stage lung tumor or a developed lung tumor that expresses NKX 2.1 and acutely deleted it? And that's the last piece of technology that I'll mention to you. A lot of our work is based on this Cre-Lox model of activation of RAS using Cre-Lox technology. Nate Young in my lab made a sister mouse which uses flip-frit recombination. So it's a frit-stop-frit version of this. Works the exact same way, except you use flip recombinase. And that's nice because it leaves Cre in your pocket to do something else with. And in this case, what we want to do is delete some other gene at some other time. So the experiment, the last experiment I'll show you, is to take the frit-stop-frit KRAS model. Your favorite gene is floxed. We also have that ROSA26 Cre-ERT2 allele to give us tamoxifen-regulated Cre. If we add flip recombinase, RAS will be recombined in those cells in the lung, but your favorite gene will still stay intact because it's floxed. That will allow a tumor to form, and then we can add tamoxifen, activate Cre, get rid of your favorite gene, and ask what happens acutely to the inactivation of a gene of interest. So in our case, our favorite gene here is NKX 2.1. We have the frit-stop-frit KRAS allele and the ROSA26 Cre-ERT2. NKX 2.1 is floxed. We add flip recombinase, and this results in a tumor forming. And that tumor, at the early stage, will still have NKX 2.1 expression. And then we can add tamoxifen. And in 7 days, NKX 2.1 is gone. It works remarkably well. You can efficiently eliminate a gene of interest in an established tumor. And then the question is, what happens to those tumor cells? What is their fate? And what happens is actually quite remarkable. And I don't know whether you can appreciate this, but the cells change dramatically within that 1 week time. The cell shape changes, the nuclear shape changes, the cell junctions change, and the entire gene expression patterns of these cells change. And what they change into is gut. The cells go backwards. They came from gut embryologically, and when we get rid of this master regulator of differentiation in the lung, they revert to an N-1 state. So there seems to be a latent program of differentiation present in these cells, and when you deprive the cells of this regulator, which is enforcing the first differentiation program, they adopt an N-1 state. I mentioned this story to you partly because of the technology, but also to reinforce the notion of the complexities of the biological circuits that are at play, even in established differentiated cells. So I'll stop there and summarize by just saying that we have in the mouse modeling of cancer community many different tools and genetic engineering tricks, which it seems to me might be well suited for collaboration with the synthetic biology community. This work was contributed to by a number of individuals, past and present from my lab shown here, as well as important collaborators. Thanks very much.
Great. We'll open it up again for questions. We have a few minutes.
Thank you for the interesting talk. We kind of noticed about the KRAS, then P53. It's very interesting. Kind of my question is, it seems the cells are happy kind of proliferating with KRAS, but there's some sort of kind of transition to adenocarcinoma that triggers the P53-mediated apoptosis. So I'm just kind of wondering if you can speculate. I can think of 3 potential kind of causes. One is maybe it's accumulating mutations or epigenetic modification. Second is, and there's some recent paper on P53 actually respond to the microenvironment, maybe something like that. A third is, or even like metabolism related, hypoxia. And third, maybe it's, EMT related. Maybe if the cell try to invade, then it will trigger something P53 related. Yeah.
So I think there are lots of possible explanations for why the cells become sensitive, but there's one simple explanation, which is that the signal coming from RAS increases at that transition. And we know that's at least true. We've actually seen the gene gets amplified between benign tumor and advanced tumors, between adenomas and adenocarcinomas. And that increased signaling leads to the expression of a P53 regulator called ARF. The adenomas don't express it, presumably because the signal is too low. The adenocarcinomas do express it because the signal is higher. If P53 is off, it doesn't matter in either case. But if you put P53 back on, it only gets activated where ARF is activated. So that relates to this interesting P53 circuit here. The other things that you mentioned could well be relevant as well, but that's certainly relevant.
I'm sorry, second question, just
Sure.
if it's allowed. So also you kind of mentioned that when you reconstitute the miR34A, you actually kind of have achieved a similar effect as kind of reconstitute P53. And my question is that, P53 has miR34 related functions, right? Targeting Notch, targeting BCL, MYC, all that stuff. But then also P53 has miR34 independent function.
Yeah.
So is that suggesting that maybe the miR34 independent function are not as important as DNA repair?
Yeah, I wouldn't go that far. I think you're right that the response that we see is similar. We see a cell cycle arrest response in both situations. With P53 restoration, actually, we see the elimination of those arrested cells through an innate immune response. We actually don't see that with the miR34 overexpression, so P53 is probably doing something to signal to the innate immune system that miR34 can't do. So that's interesting in and of itself. And also we're expressing miR34 probably at very high levels compared to what happens physiologically. So it might be that in a normal P53 response, miR34 contributes, but it may not nearly be sufficient in a normal physiological response. And those other components probably, almost certainly, are contributing as well.
Another question?
Tyler, with regard to the last story you told, it seemed like you had a pretty good hypothesis for NKX 2.1 playing a role. I'm wondering, with the system you have set up, how easy it would be to test a library of your favorite genes, either from studies, GWAS studies of cancers, or just all the genes to see what might give metastases.
I think any gene that you have in that flox configuration you can do. It's not a trivial experiment, right? You have to do all the breeding and so forth and so on. I think it would be better to imagine modifying the system to allow you to do it in a more high throughput way. I wouldn't do it by germline mutagenesis, as we've done in that example. There are other ways of doing it. In fact, Anton Burns is pushing the idea of making ES cell-based versions of these models, where you don't have to do the whole germline breeding thing. You just make ES cells with the relevant changes. And maybe using CRISPR technology, for example, you could make your mutations more rapidly in an ES cell context, and then put them into chimeric mice and do the same experiment there. That would be a much faster way. But in principle, using the tools that we have, you could look at any gene of interest. Yeah.
Great. We'll take two more questions. Go ahead.
So there are obviously many different populations of cells within the lung, and my understanding is also that there's been a lot of discussion on whether there's a stem cell in the lung. When you infuse, it's interesting, when you infuse the adeno and lenti, you have the potential to infect perhaps a good number of those. Which cells give rise to the tumor?
Yeah, you want to come to my lab? It's something that we've wondered about for a very long time, and the answer is likely to be more than one cell type can do it. And that comes a little bit from our work and more from this guy I just mentioned, Anton Burns in Amsterdam. He's used viruses that have cell type specific promoters, and he can get tumors that look like these from a variety of different sources. So I don't think it's just one. We think that there's a stem-like cell that is particularly good at it, but I don't think it's the only one that can do it.
Please.
In the last segment, you showed that there was a reversion to the gut-like. Is that true for the metastatic sites as well?
We haven't done it in that setting. We've only looked in the primary tumor, and it would be hard to do it in the metastatic setting because there, NKX 2.1 is epigenetically silenced.
Right.
It's already off. So it would be actually, you'd be going from off to off, and it wouldn't be clear what to expect there. So we've only looked in the primary tumor where it starts off being expressed and then eliminate it.
Great. Well, thanks, Tyler. Thanks a lot.